All right, let's just jump right in. I want you to picture the most complicated instruction manual you can possibly imagine. Now, what if that manual was for you? Because that's pretty much what our DNA is. It's this incredible set of instructions we get from our parents, passed down generation after generation.
And this manual, I mean, it dictates just about everything. We're talking about stuff from your blood type, all the way to something as fundamental as your biological sex. It is literally the blueprint that makes you, you.
You know, for most of human history, that manual was a completely closed book. We were just stuck with the code we were given. End of story. But now, we're learning to read it, copy it, and yeah, even rewrite it. So that brings us to the biggest question of all. What happens when we start hacking our own biology? Okay, so before we can even think about rewriting the code, we've got to understand the basic rules of the game. So let's start at the beginning. How we inherit some of our most fundamental traits. It really all comes down to our 23rd pair of chromosomes. See, females have two X chromosomes, so all of their egg cells will carry a single X. Simple enough. Males, on the other hand, have one X and one Y, which means their sperm is split 50-50. Half carry an X, half carry a Y. This means that at the moment of conception, determining biological sex is basically a coin toss, a 50% chance either way. Now, blood type, that's a little more complex. It all has to do with 3 different versions, or alleles, of one gene. Think of the A and B alleles as being dominant. If they're there, they're going to show up. The O allele is recessive, so it only makes an appearance if you get 2 copies of it. But here's the cool part. A and B are codominant, so if you get one of each from your parents, you don't get one or the other, you get both, and end up with AB blood type. So understanding these rules was the first big step. But for thousands of years, humanity has been trying to, well, bend those rules to our will. And we've been using our oldest tool for genetic manipulation to do it, selective breeding. And the problem driving all this was simple, and it was huge. As the human population just kept growing and growing, we needed more food. We had to figure out a way to make our crops and our livestock more productive. If we didn't, we'd starve. The solution was, patient, really deliberate. It's a slow process. First, you decide what trait you're looking for. Let's say a cow that makes more milk. Then you find the male and female who are best at that and breed them. From their offspring, you pick the best of the best and do it all over again. And again. And again. For generations, until that trait you want becomes the norm. And honestly, the results have been absolutely incredible. This is how we got disease-resistant crops, wheat that gives us tons of grain, animals that produce more meat, and even how we shaped our pets, like breeding dogs specifically for a gentle nature. I mean, almost everything on our farms and even in our homes has been touched by this process. But, and this is a big but, there's a serious downside. When you focus on just a few genes you like, you shrink the gene pool. You reduce genetic variation. And that makes an entire population of plants or animals super vulnerable to a single disease. Just think about the Irish potato famine. It was so devastating because there was basically no genetic diversity in the potato crop. Sometimes you also accidentally select for a health problem that's linked to the trait you want, like hip problems in certain large dog breeds. It's a powerful tool for sure, but it is definitely a double-edged sword. You see, selective breeding is kind of like carefully shaping a piece of wood with a chisel. It takes a long time. But today, we've traded that chisel for a power drill. So let's look at the modern toolkit, which is way faster, way more precise, and, well, far more controversial. This right here is genetic modification. Instead of waiting for generations, scientists can act like tiny microscopic surgeons. They use these things called restriction enzymes. Think of them as molecular scissors to snip out one specific gene they want, maybe for drought resistance. Then they use a carrier, like a harmless virus or a bit of bacterial DNA, to deliver and paste that gene right into the code of a whole other organism. And the potential here it's just enormous. People who support it argue that this is one of the keys to solving global hunger. We could grow crops that are more nutritious, that can survive tough conditions, and that produce way more food. We're talking about feeding millions of people. On the other hand, you have some really significant ethical concerns. A lot of people wonder about the long-term consequences of messing with the fundamental code of life, of moving genes between species in ways that would just never ever happen in nature. All right, another powerful tool in this modern kit is cloning. And no, this isn't science fiction anymore. We're talking about making a genetically identical copy of an organism from just a single parent. Of course, the most famous example was Dolly the sheep, who was the very first mammal to be cloned from an adult cell way back in 1996. Now, when we say cloning, it's not just one thing. Gardeners have been cloning plants for centuries just by taking cuttings. Tissue culture is a much more high-tech way of doing the same thing. But it's that adult cell cloning, the Dolly method, that brings up the most complicated ethical questions, especially when we start talking about animals. So beyond changing the genes themselves, we've also engineered the entire environment around our food. Intensive farming is just a whole system of trade-offs. We use herbicides to kill weeds so more energy goes to the crops, but that tanks biodiversity. We keep animals indoors to maximize their growth, but that confinement can lead to diseases spreading like wildfire. It's this constant balancing act between getting the most food possible and dealing with the consequences. So we've learned how to select for traits, how to modify genes, how to copy them, mostly for our food. But what about the most complex, most important code of all, our own? And that brings us to one of the greatest scientific projects in all of human history. 3 billion. Just let that number sink in for a minute. That's how many base pairs there are in the human genome. And from 1990 to 2003, this massive global team of scientists worked to sequence every single one. The scale of the Human Genome Project is it's mind-boggling.
And the payoff? It's been immense. Having this complete blueprint means we can now hunt for specific genes linked to diseases, like the BRCA1 and BRCA2 genes connected to breast cancer. It helps us understand and treat inherited disorders, and it even lets us trace the migration patterns of our ancient ancestors all across the globe. From simple observation to direct hands-on intervention, we have built an absolutely incredible toolkit for messing with the code of life. So, where does that leave us now?
So let's quickly recap what's in our toolkit. We have selecting, right? The slow and steady work of our ancestors. We've got modifying, the fast, precise, but controversial new power. We can create clones, perfect genetic copies, and now we can read the entire blueprint from start to finish.
And all of that leaves us with the ultimate question, doesn't it? For the first time ever, we have this incredible, almost godlike power. We are no longer just subject to our genetic inheritance, we're becoming its editors. The science is moving at a breakneck pace, but the ethical questions are still right there with us. So, what's next? What should we do with this power? That right there is the code that all of us have to figure out together.